Excitation-contraction coupling is the process by which smooth muscle contracts in response to a stimulus. It is the state of smooth muscle contraction that determines the diameter and tone of blood vessels. This in turn regulates blood flow and affects arterial blood pressure. You will see how vasoconstriction, or narrowing of blood vessels, is brought about by smooth muscle cells contracting, and how vasodilation results from smooth muscle cells relaxing. We will cover the mechanisms that lead to smooth muscle contraction and relaxation. Excitation-contraction coupling can be regulated by the membrane potential, such that depolarization evokes contraction. This is known as electromechanical coupling. But smooth muscle can also contract independently of membrane potential. When depolarization is not the trigger, it is referred to as pharmacomechanical coupling. This photograph shows smooth muscle cells that have been isolated from an artery wall. You can see that the cells are long and skinny. This fluorescence image of an arteriole shows how the muscle cells are oriented around the vessel wall. The blue stain shows individual cell nuclei, while the green stain is due to a voltage-sensitive dye and it's brightest around the cell membranes. So you can distinguish individual smooth muscle cells in the vessel wall. For example, there is one here, which interlinks with another cell outlined here in red. It disappears around the other side of the vessel. A large part of this cell, outlined in blue, spans much of the width of the artery. The orientation of the cells gives rise to a clear stripy pattern that wraps around the circumference of the vessel. The cartoon at the bottom illustrates the consequence of this structure. As the muscle cell contracts and shortens, the vessel lumen becomes narrower, showing constriction. In contrast, as the cells relax and become longer, the vessel lumen dilates. As with other types of muscle, the key mediator of contraction is the interaction between actin and myosin to form cross bridges. In smooth muscle, this interaction depends on the phosphorylation of myosin, more specifically the myosin light chain. The state of phosphorylation of myosin depends on the relative actions of two enzymes. Myosin light chain kinase promotes myosin phosphorylation by transferring a phosphate group from ATP. The removal of phosphate from myosin is mediated by the enzyme myosin phosphatase. So what determines the effects of these two enzymes? Contraction begins when there is a rise in the concentration of free calcium ions in the cell cytoplasm. The cells contain the calcium binding protein calmodulin, which interacts with calcium to form a calcium calmodulin complex. This complex in turn activates myosin light chain kinase to phosphorylate myosin. A number of signaling pathways can interfere with the reaction between calcium calmodulin and myosin light chain kinase. For example, cyclic AMP and cyclic GMP both inhibit the reaction and cause relaxation of the muscle. Many vasodilators act via the cyclic AMP or cyclic GMP pathways. At the other side of the equation, myosin phosphatase activity is inhibited by Rho kinase and by protein kinase C. These enzymes are activated by a number of vasoconstrictors, which promote smooth muscle contraction. This mechanism means that although calcium is needed to trigger contraction, it is not needed to maintain the contraction, and contraction can be sustained with minimum consumption of ATP. Thus, blood vessels are able to maintain constriction for prolonged periods. Since calcium ions are the trigger for contraction, it is important to understand how the cytoplasmic calcium concentration is regulated. We will focus first on electromechanical coupling, whereby the membrane potential regulates contraction. This is a cartoon of a smooth muscle cell. The cell membrane contains a variety of ion channels that contribute to the control of the membrane potential. The key channel for contraction is the L-type voltage-operated calcium channel. When these channels are open, calcium enters the cell and directly triggers contraction. Calcium can also enter the cell through cation channels. Several types of cation channel are expressed in vascular smooth muscle. Some are receptor-operated, such as P2X channels and members of the TRP superfamily of channels. Others are store-operated, such as channels formed by the interaction of ORI1 and STIM1 proteins. I won't go into the mechanisms of activation of these channels, but suffice it to say that they open in response to the depletion of calcium stores in the sarcoplasmic reticulum. Some cation channels have substantial permeability to calcium, and enough calcium can enter the cell through these channels to directly trigger contraction. Most cation channels are more permeable to sodium than calcium, and the main function of cation channels is therefore to mediate the influx of sodium ions, which depolarizes the cell. The depolarization, in turn, activates voltage-operated calcium channels and calcium influx. Other ion channels affect calcium influx by changing the membrane potential. When chloride channels open, chloride ions flow out of the cell, causing depolarization, which enhances the activation of voltage-operated calcium channels to cause calcium influx. When potassium channels open, potassium ions flow out of the cell to cause hyperpolarization. This counteracts depolarization to close the voltage-operated calcium channels and inhibit calcium influx. Positive and negative feedback mechanisms also contribute to the control of membrane potential and calcium influx. As the calcium concentration rises in the cell, calcium can act back on chloride or potassium channels to cause activation. Depending on the location and relative density of calcium-sensitive chloride and potassium channels in the cell, the overall effect could be depolarization or hyperpolarization to further promote or inhibit calcium channel activity and calcium influx. As a consequence of the key role played by voltage-operated calcium channels in regulating calcium influx and contraction, drugs that inhibit the activity of calcium channels are effective vasodilators. There are three chemical classes of calcium antagonist: the dihydropyridines, the benzothiazepines, such as diltiazem, and the phenylalkylamine, verapamil. They all inhibit the L-type calcium channel, but they show differential selectivity toward calcium channels in smooth muscle compared with heart. The dihydropyridines are more potent in smooth muscle than heart. The dihydropyridines are therefore the class of antagonist that is used clinically to treat hypertension and angina. Smooth muscle selectivity arises because the drugs interact preferentially with inactivated calcium channels, and smooth muscle is substantially more depolarized at rest than cardiac muscle. A higher percentage of calcium channels are therefore inactivated at rest in smooth muscle, which increases their affinity for dihydropyridines. Nifedipine was the first dihydropyridine to be discovered, but there are now a lot of drugs in this class. A few examples are shown here. Amlodipine is the most widely used clinically due to its better pharmacokinetics. In pharmacomechanical coupling, the cytoplasmic calcium concentration rises as a consequence of a substance activating a G protein coupled receptor, which uses the GQ-G11 family of G proteins to transduce the signal. Upon binding of an agonist to the receptor, the G protein is activated and the G-alpha subunit stimulates the activity of the enzyme phospholipase C. That results in the cleavage of phosphatidyl inositol to IP3 and diacylglycerol. IP3 diffuses to the sarcoplasmic reticulum, which acts as an intracellular store of calcium. The IP3 binds to and activates the IP3 receptor, which is a calcium-permeable ion channel in the sarcoplasmic reticulum membrane. Calcium ions flow out of the sarcoplasmic reticulum down their electrochemical gradient to stimulate contraction. The diacylglycerol that is generated alongside IP3 causes calcium sensitization due to inhibition of myosin phosphatase, as illustrated earlier. The two branches of the pathway therefore work together to trigger and sustain contraction. Examples of G protein coupled receptors that mediate contraction in this way include the alpha-1 adrenoceptor, angiotensin, vasopressin, endothelin, and serotonin receptors. I've so far only described pathways that raise calcium in the cell. Mechanisms that remove calcium from the cytoplasm also play an important role in regulating the free calcium ion concentration. The predominant route is calcium reuptake into the sarcoplasmic reticulum, which is mediated by the sarcoendoplasmic reticulum ATPase, or calcium pump, known as SERCA. A separate calcium pump, the plasma membrane calcium ATPase, or PMCA, pumps calcium out of the cell to the extracellular space. Calcium is also removed from the cell in exchange for sodium ions via the sodium-calcium exchanger.